This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, recorded on July 23, 2020. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello there. Hello, everybody.
How are you doing? Are you still sheltering?
We're sheltering. We are locked in like crazy, but we're doing all right. I live in a retirement community, and so everybody else is in the same boat. But we manage.
All right. Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Are you still sheltering? No, you're going to work, right?
I'm going to work, sort of, kind of. I'm working from home and going into work only like you are occasionally to do things. Tomorrow I have to go in and film a video for a clinical trial that we're doing, and we're doing the instructions for the study subjects so they know how to accomplish a point-of-care serology test that we're asking them to do.
Our guest today is coming back to TWiM for the third time. He was previously on 180 and 201. He's from the University of Puget Sound. Mark Martin, welcome back.
Thank you, Vincent and Twimmers.
Nice to have you.
Oh, it's delightful. And I've been trained. Current temperature today in Tacoma is cloudy and 59 degrees Fahrenheit, 15 degrees C, and 288 degrees Kelvin.
Very good, Mark.
Now, that's the Twivers. That's the Twivers.
It's pretty chilly.
So we'll just call ourselves Twimmers is what I meant to say. See, I've been listening to Twiv too much.
Well, you can't listen too much, can you? Now?
That's right.
You can never listen too much.
That's pretty mild out there. Is that typical for July?
Yeah, our hot spell was 84 degrees yesterday. And as Elio will tell you, since I'm born and raised in the Los Angeles or Southern California area and lived in San Diego for 10 years, it's very different there.
It is indeed.
Now, Mark, what are you up to these days? Are you going into your office yet, or what? What's the story?
So we have permission to the statement that I make is we need to stay masked, distanced, and positive, but not in the virological sense. And so I have one research student, undergraduate, because that's all we have at the University of Puget Sound, and we stay 6 or 7 feet apart, and I usually go in the morning, and my student comes in the afternoon, and everything else is generally done via Zoom, which I should have invested in.
You bet. Who knew? Are you going to have classes in the fall?
We will know next week. I believe I have shared with you the pretty close to a spacesuit that my wife, Jennifer Quinn, purchased for me, which is amazing. Has N95s in and out. It makes my friends so crazy to look at, but it's my dream. But to answer your question, we've been trying to go forward with the idea of teaching face-to-face, which, to be very honest with you, seems like a dodgy proposition at this stage. It's true the Department of Health here in Washington State, I mean, our case numbers are going up, but hospitalizations stay level and decreasing. And of course, our deaths, thank goodness, are very, very low. But when you bring in a whole bunch of students, the statement was we would test everybody when they arrive and keep doing it, but not the way Michael Minna taught us.
Yep.
So I imagine, I mean, for me, and I'll keep this short, I apologize, is that I don't know how I can do a really great job at a small liberal arts college teaching the laboratory part of microbiology. So this has been my sleepless nights trying to figure that out.
Yeah, I'm sure. Well, you're in the state where it all started, really. Virus first come in Washington.
It's all
right?
your fault.
It is. It is.
That one guy who flew in from Wuhan.
Yes.
Back in January, I guess.
Patient zero.
Yeah. Well, thank you for joining us, Mark. And It's always good to hear you. The previous times were both at ASM Microbe. We And the latest one was last summer, right? 2019 in San Francisco. That was episode 201. All right, so Mark is going to start us off with a snippet. And before we do, I just want to remind everyone, if you like what we do on all of these podcasts, you could support us financially, microbe.tv/contribute. Mark, what do you have for us today?
Well, the first thing I have to do is apologize to Michael that after listening to last week's TWiM, I don't have fecal material or mucus to discuss today with you.
Oh,
I do. But I do have microbial dark matter, the dental microbiome, parasitic/predatory bacteria, and Neanderthal calculus to discuss.
that'll make Elio happy.
Yes. And I promised to my good friend Elio we'll keep to 20 minutes on this.
That's great. I'll hold you to it.
Oh, I hope so.
He will.
I hope so. So it's from Cell Reports, and let me check the date. I know it's 2020, but it looks like came out July 21st, 2020. So this is fresh baked. And the title of the article is Acquisition and Adaptation of Ultra-Small Parasitic Reduced Genome Bacteria to Mammalian Hosts. And there are a sizable number of authors, including Jeffrey McLean as lead, along with Batlib Bohr, Christopher Cairns, Quanhui Liu, Tao Tao, Lindsay Solden, Eric Henderson, Kelly Wrighton, and Weihan Shi. Oh, and Zisong He. I hope I did that correctly. They're at the University of Washington up in Seattle, not far from here, and some lovely additions from the Forsyth Institute, which does great work in Cambridge, Ohio State, and back to the Harvard School of Dental Medicine. And the first thing yes. And the first thing I need to tell you is I really, as an educator, as well as a very low-level researcher, I very much enjoy the fact that many journals have graphical abstracts to help you with things. And so the first page of this is a beautiful graphical abstract that pretty much shows the whole story. That basically, and I'm going to say this very briefly, that this paper shows that humans are inhabited by a broad diversity of these nano-sized bacteria. No, I don't mean the NaNNO bacteria that some of you might remember. And they're within a new grouping they propose to call the Sacchari Bacteria. And they are related to this beautiful, the candidate phyloradiation group that I remember being confused about when I was teaching about it. But what's interesting in the figure next to it is it's very clear that these are extremely common microbes. And they seem to appear in sea mammals, ruminant mammals, sludge bioreactors, groundwater, ancient dental calculus, and you and me.
Who would have thought, huh?
I know. And I have to say, I love predatory microbes, as most of you know, and there are lots and lots of mechanisms for this, ranging from, and Elio, you wrote about this on Small Things Considered, ixotrophy, this business of getting tangled up in flagella, to the beautiful wolf packs of the myxobacteria. Lynn Margulis's work really informed me about this years ago, and I think one of the things to consider when we talk about this aspect of the paper is the possibility of stealing nutrients from other microbes is a winning strategy, and we're going to see it often. The whole concept of microbes interacting with microbes, which sociomicrobiology it's often called, is everywhere. So with that, I wanted to say that when I actually have my undergraduate students, juniors and seniors, discuss a paper with me, I often find another version of it, usually an editorial or a short version of it, and I'll include in the show notes something from Science Daily so you can compare and contrast the way that I did, the kind of business in there. Some of them are more trustworthy than others, as Elio laughingly knows. So the first thing I want to do is I want to talk about microbial dark matter, and it's one of those scientifically attractive terms that means very little. I would say that we're limited by our techniques to analyze what's there, and as our techniques improve, so does our vision. And so this business of microbial dark matter bothers me a little bit, but it's certainly true. We're always finding out new things. And I remember teaching a little bit about an organism that was called TM7 for quite some time. Now, this is a mysterious sort of taxon related to what are called the candidate phylum Radiation. And it's really interesting when you have a look at that at the paper to see, and I'm looking actually right now at figure 1A, that there are a lot of these things, but we know very little about them. And so the grouping that we're discussing today, which are associated with the CPR, are the Sacchari Bacteria. Now, about 5 years ago, one of the co-authors of this paper actually isolated one member of TM7. And when I say they could cultivate it, and Elio, you're going to love this, it's not really isolated in the sense that you and I might mean. You can grow this in the presence of its prey organism on, and Michael, this is for you, SHI medium, which is typical with protease peptone and some other complex things, with added human saliva.
They're supplying the necessary growth factors that it probably is not getting. The necessary growth factors it's not getting from the complex medium.
I agree. And it's interesting on top of that, Michael, because you can't grow this by itself. So in other words, you have to make sure you can grow the quote, prey organism. They like to use this term parasite, which I'm a little uncomfortable with. Elio, do you remember Mortimer Starr?
Oh, yes.
Mortimer Starr wrote a whole review paper in Annual Review of Microbiology about whether or not my good friend Delovibrio was a parasite, a parasitoid, or a predator. It was 26 pages or so. So yes. I don't know what to say.
It's a predator.
Yeah.
Delovibrio is a predator.
Well, that's how I feel.
But you cannot grow Delovibrio by itself, right?
No.
Well, that's another topic for another time, and I would be delighted to. You can grow host-independent mutants, but that puts a burr.
You're cheating now,
right? But that puts a burr under my saddle as a microbiologist. So 5 years ago, one of the co-authors, Zay, showed that this organism, TM7X, grew as an epibiont on an oral actinomyces.
And in the abstract, in the graphical abstract, it almost looks like that fish that attaches itself to a shark.
As a mora.
But you know, as I first looked this paper up when I was invited to join you folks, it reminded me, and it was so gratifying to me that I thought this, and then I read about it. So in other words, I'm actually up to date on a lot of things. It reminded me very much of Carl Stetter's work with nanoarchaeum and ignicoccus.
That's right.
Where you have these small spheres that attach epibiotically. And I just love this. And, you know, that's between two different archaea. And this is between two different bacteria. Now, you do get clear lysis of the host bacterium, and the microbial beast, our little friend TM7X, is extremely reduced. It only has 707 genes, they're predicting. It cannot make any amino acids or vitamins. And the proposed name that they give for it, I wish I could name things. This is cool. Nano-zymbacter lyticus.
So it tells you what it does.
Yeah. Isn't that nice?
It's small, and it lyses cells.
And this thing is small. And Elio, I'm curious what you think. It's estimated to be between 200 and 300 nanometers in diameter. And that makes me wonder if all this central dogma machinery is shrink-wrapped. Because I'm trying to figure out what the volumes of the enzymes and ribosomes and such are.
It's not volume, it's the number.
It just doesn't have a lot of ribosomes, for sure. And, it's got a tiny nucleoid. I mean, small is relative. But this is about, a lot has been written about what is the minimal size in biology. And 200 nanometers is about it. So this is about as small as it can get theoretically.
It's very interesting because one of the things, and I don't know if you remember this, Elio, you spoke to me years ago about, are the ultra microbacteria. Marine bacteria, when they're starved, get very, very small.
Right.
And that's where I first came up with this like image in my mind of being shrink wrapped around all the machinery of the cell.
Sure.
But this is going to be very...
In fact, Banfield makes the point that, I think she makes the point that most bacteria in the world are very tiny.
Yes.
When we think about the average bacterium being E. coli, that's wrong. E. coli is way too big.
It's one of the giants.
That's right.
So this is a problem that I teach my students. I say the danger is coli centricity.
Right.
Where we think everything's like E. coli. Same thing with eukaryocentricity and oxycentricity.
So let me interrupt because a few weeks ago, my son asked me, why do people work on E. coli? And
it's Elio's fault. It's Elio's fault.
He didn't like my answer. So,
oh my God.
Why are we working on E. coli? Is that Josh Letterberg's fault?
No, it happened before. What happened was that E. coli was easy to grow. The strains in the lab are not pathogenic. And so you accumulate knowledge. And when you accumulate knowledge about an organism, it becomes a model organism because there's more known about it. You know how to handle it. You know how to manipulate it. So it became the king by attrition, by adding knowledge to it. But there is a candidate now. There is a tiny little bacterium. What is it called? You know, Mark, you would know.
Do you mean Vibrio natrenges?
Yeah, that's what I mean.
Yeah.
That's supposed to take over from E. coli because it's got every advantage. It grows much faster than E. coli, for instance.
It does. And it's naturally transformable. And I got some great information on trying to transition that into my classes. So I'm really hopeful about that one. I like this kind of stuff.
And it's not pathogenic because unless you're a fish.
I don't think it's even for fish, Michael, but I could be wrong about that. That's interesting, though. So so this is a very interesting organism. There's no work that suggests breadth of prey. In other papers associated with it, the authors suggest that maybe it's only after that actinomyces. But I question it because the process of cultivation modifies the organism. I say like the hand of Darwin is on all that lives. So evolution is taking place all the time, right?
Well, let me ask you this question. Given the fact of the cellular morphology of the actinomyces, they make this filament. Could this be just a straw that this TM7 attaches to and literally sucks the life out of the actinomycete?
Yeah. It reminds me of an organism that Lynn Margulis wrote about that I do not believe can be cultivated called Vampirococcus.
The vampire bug. Yes.
Right. And you know, I was always a fan of the one that just barged into other cells called Daptobacter, but I don't believe those are cultivable yet. But I think you're right. There may be something about the geometry of what we're looking at because later on in this paper, when they start to look for prevalence of relatives of TM7X using fish, they basically saw small round targets attaching to long rod-like organisms. So you may be onto something, Michael.
It's the surface to mass ratio because you're able to effectively sneak in without violating the pressure tight seal of the microbe and be able to pull things out.
You know, that's something that fascinates me and always has about the biophysics of this. If you start thinking about, for example, Della Vibrio entering the periplasm of a gram-negative, that's 40 nanometers wide.
Mm-hmm.
So that's one heck of a molecular shoehorn. And we know it's a type 4 secretory system that helps with that. But still, I would love to see more, and maybe with more of the, oh my goodness, freeze fracture electrotomography, we can start to look at the machinery that actually puts that soda straw into the organism, Michael, which would be fascinating. So, again, what I call a Darwino genome, it's become very reduced. And it turns out that when they start looking in metagenomes, they find it very common. These relatives of this organism are extremely common, found not just in us, but in ruminants, in groundwater, and my good friend sludge bioreactors. Right?
Can't live without sludge. We need sludge.
I repeat myself. Who would have thought?
Yeah. When I took the microbial diversity course, there was an instructor who refers to the sludge as being black gold, which I thought was great. So I mentioned before with taxon-specific fish microscopy, you can show this kind of association with relatives of TM7X pretty much everywhere. And I don't know why they did this, because it wouldn't have occurred to me. Turns out they had some Neanderthal calculus, tooth plaque.
As you would at the Foresight.
Yeah. And so it turns out it's in there too. And that's from their estimate 48,000 years ago. Now, I believe they're looking at this to establish whether what we're seeing are recent or older events. But again, timescales, and Elio hit on a really important point a moment ago. We often think about fastest is best, but the majority of organisms are small and slow growing. And we don't think about that enough. So they found it fairly commonly, and they actually wonder, and this is something that's relevant to some work I'm doing in the lab, that when you try to estimate numbers of things in a metagenome or a large library, and you base this on the numbers of copies of 16S ribosomal RNA, that little operon, many organisms, Tom Schmidt showed this many years ago, they have multiple copies of these things, like spore formers, for example. But this just has one. So it could be this is even more.
That's kind of unique, isn't it?
It is.
I know of no other bacterium that only has one copy of 16S.
I kind of want to say that there's an intracellular pathogen that does, Elio, and I want to say Legionella, but I could be wrong. I wish Michelle were here. I know, we're all going to look it up. So what was interesting is when they start looking at these members, again, that they're not growing, this is just in silico work, they found that there were lots of similarities to other microbes in this CPR grouping that are dependent on other things. And they start looking at the genes involved with that, and they found things like, it would make Steve Finkel happy, DNA uptake mechanisms. There are some obligate amoeba symbionts that seem similar to this. And every one of these has type 4 secretion systems. There's even some talk about maybe working Sec-A backwards, something that Milton Sayer talked about many years ago with me. So that's all fascinating to a person like me. But to someone interested purely in evolution, you have to ask yourself, what can you say about this process of becoming intimate with humans, which is what we're talking about? And it turns out that the environmental genomes and the host-associated genomes are extremely similar, suggesting only a few changes are necessary to transition from being, say, in groundwater to being between my teeth right now, which is fascinating. They finally suggest that maybe we should consider this as the equivalent of a polyphyletic event. It's happening several times. And I've often wondered if some microbes are kind of preset for these kinds of associations. And the final thing I'd like to, because they did not mention it in their paper, and I'd like to mention it to you three and the listener, is that this reminds me so much of the Thor association work done by Jo Handelsman and her colleagues at Madison. Now, these are cytophaga gliding bacteria that live within the peptidoglycan of B. cereus and actually use elements of it. I used to call them peptidoglycan surfers, but I didn't think Jo liked that.
She probably didn't.
No, but she tolerates me, so that's good. John Donne wrote that no man is an island, and I don't think microbes exist alone. And this is a great example of it, because I don't know that every association we're going to look at like this is necessarily parasitic or predatory, but it would be a short hop to get there, to go from a surface to a source of nutrients. So just to sum up, the highlights of this whole paper, and I always tell my students to come up with some take-home lessons, And these Sacchari Bacteria, which is the general grouping, are ultra-small parasitic bacteria recently discovered in humans. Turns out that there is a lot of diversity within mammalian hosts. You find them all over the place. They generally are ultra-small, reduced genomes, less than 1,000 genes in a single 16S copy. One of the groups, the one that they're studying most, shows minimal changes since going from the environment to humans. And it's interesting that they don't really talk that much about predation in this paper, but they do in a previous one.
So let me ask you this, Mark.
Certainly.
Why are they there, and what are they doing? The two questions for which there are probably no answers. I think they're there because it's the whole microbe-host interaction. And these very small bacteria may be taming the wild beast of the normal bigger bugs that we have in niches. Effectively, as they're fighting off the predator that's trying to suck the life out of them, they're not messing with us by throwing out signaling or, building massive amounts of biofilm. And it's just part of this intricate dynamic. And so I guess my question to you is, should we begin to build into our bioinformatic questions when we look for the importance of the microbiome and changes in the condition? Should we build in this phyla to see what their numbers are doing in the context of disease or health or whatever question we have to be asking?
You're not asking a question. You're making a statement. The answer is, of course.
Well, I have a tendency to go on. Michelle normally reins me in.
There's another thing here. When you think about fulfilling Koch's postulates, you have to put something in an animal, cultivate it and put it in an animal. When you put it in an animal, what happens is it picks up nanobacteria. So Koch's postulates are a little bit more, they have to be expanded a little bit to think that it's not just the bug, it's the bug and its friends, the nanobacteria.
Yeah.
So Elio, with real estate, they say location, location, location.
Right.
And I often say in microbiology, it's context, context, context. And this is how do you define a pathogen? I've been through that quite a bit with Arturo Casadevall. He actually suggests we should call them pathogons, which is pretty funny. But it all depends on the associations you're talking about. And this is one of the things that fascinated me about nanoarchaea and ignococcus, is whether or not it was an issue of a mutualist or a predator, and it's really hard to study those. Well,
it's not. You used to think that this was an exception. In fact, it's going to turn out to be the rule.
Yeah.
I think so. I make a joke about it. There are 3 stages to scientific research, and the first is that's a dumb idea. The second is it might work a little bit in the lab, but it's not relevant to the natural world. And the third is I said it was a good idea all along. And I've seen that happen with bacteriophages, which I always suspected were drivers of ecology. And I would argue that predatory bacteria, maybe that's just a subset. Interactive bacteria might be a better way of putting it. Are real drivers.
It's a much better term.
They're really, really involved in shaping communities.
Yes.
And so it could be that some of the examples they talk about here, very similar to Joe's system, could be used to start dissecting these kinds of associations.
Beautiful. I'm, as always, Mark, I'm always pleased, excited to see how much insight you have into various aspects of microbiology. We are a walking encyclopedia.
It's what they called me in elementary school.
And Mark, can we find the ancestors of these guys?
Yeah, I believe we can. And I think that if you look at some of the stuff in groundwater in particular, where they put a lot of their focus, I would imagine that would be the place to put. And what I would do, and I am not them, is I would try and bait groundwater with this one particular known prey and see whether or not, because it's all about delta G. Any way that you can make a living, microbes will take advantage of.
Nice.
And, one more question for me. Besides mammals and sludge and any other animals?
They don't say so, but I'll bet they didn't look.
Yeah, probably not.
That's the way it also happens. We've all seen it in our own scientific research. You'll hear about something that you will find out something, I should say, and then you talk about it with an expert and go, "Oh yeah, we all knew about that," but no one had looked. So I can't wait to see how they follow this up. And this is great because it shows how people involved in quote, health professions can really appeal to general microbiology as we start to think about this.
Wonderful. Thank you very much.
Thank you so much, Mark.
You're very welcome.
All right, Dr. Schmidt, what do you have for us?
Well, we have something completely different, but equally as odd. We're going to talk about a sensitive magnetic arsenite specific biosensor hosted in Magnetotactic Bacteria. And this is from the Marseille University and St. Paul de Reims, France. And it's by Anis Dudonnet, Sandra Prevenal, and David Pinola. And so the paper is looking at the use of magnetotactic bacteria that we have discussed previously on TWiM, but the short bullet is bacteria have tiny little magnets in them that effectively they use to steer their motility, and it effectively helps them find the right redox zone in sediment using the magnetic force lines of the planet. So what these authors wanted to do is to use the magnetotactic bacteria that they abbreviate as MTB, which drove me crazy for the first 2/3 of the paper because I kept saying tuberculosis is involved, because MTB for me is tuberculosis, Mycobacterium tuberculosis. Every clinical microbiologist abbreviates that way, and so do the magnetotactic bacteria investigators. But they want to use the magnetotactic bugs as a cellular chassis to design and build sensitive magnetic bacterial biosensors. So why here and why now? I'm trying to give us a little break from thinking about COVID while at the same time thinking about COVID. This paper I picked because I listened to Mark Minna on TWiV and heard his discussion about sensitivity and specificity for the COVID test. And this paper really helps you think about those issues. And we'll get into that as we walk our way through the paper. But arsenic contamination of groundwater is widespread. And there are a number of regions where arsenic contamination of drinking water is really quite significant and troubling. Many of us have heard about the disaster of the groundwater wells in Bangladesh since the early 90s, and its wide occurrence in well water in that country. And because that nation is under-resourced, they unfortunately don't have access to the ability to clean up their water, to rid it of arsenic. And it's now recognized that at least 140 million people in over 50 countries have been drinking water containing arsenic levels above the World Health Organization's provisional guideline value of 10 micrograms per liter or 0.13 micromolar. And the signs and symptoms caused by long-term elevated exposure to inorganic arsenic differ between individuals, but principally chronic exposure can result in the thickening of skin. You develop a darker skin tone. You develop chronic abdominal pain, diarrhea, heart disease, numbness, and of course, the scariest of all, cancer. Mechanistically, what arsenic is doing is it's interfering with our mammalian cellular longevity by its allosteric inhibition of an essential metabolic complex, principally the pyruvate dehydrogenase complex, which every self-respecting microbial physiologist knows catalyzes the oxidation of pyruvate to acetyl-CoA through the mediation of pulling off the CO2 and taking the electrons to NAD. And when this enzyme is inhibited, the energy system of our cells are disrupted to the extent that it sends us into an apoptotic crisis. Now, treatment is available, but again, this is resource intensive and it's really chelation therapy. So it's not you give the person a pill and you pat them on their head. It's really, really pretty bad. For the pharmacologists who routinely listen to us, arsenic compounds have been used as our first antimicrobials, as they have taught us. Salvarsan or arsafine, which was first synthesized by Paul Ehrlich's group with its anti-syphilitic activity discovered or began to be widespread used in 1909 after Hatta attributed to inactivating spirochetes. So, thinking about arsenic, it's a big thing. So being able to develop a method, and I've put into the show notes the WHO's website for arsenic and arsenic contamination, as well as our CDC site, because the United States does indeed have issues with arsenic in some of our groundwater supplies. So enter our paper. I believe that our audience is well aware of the value proposition that reporter cassettes offer. Simple in design, they have been used by molecular biologists since the days of Jacob and Minot, with much of the work resulting in exploiting them as tools from the elegant work of John Beckwith in his multitude of progeny, and with there being two principal types, transcriptional fusions and translational fusions. So today we're going to talk about transcriptional fusions. However, they each share in common is that when the transcription operon senses a signal, they switch from the off to the on position, which the end user actually can visualize very easily. In the early days when I was being trained, the reporter was the principal end product of the lac operon. And that's, of course, what Jacob and Minot and Beckwith made their bones on. They worked beta-galactosidase brilliantly. And many of us remember using ONPG or its more simple reagent, XGAL, or as its trade name was, Blue Gene, which was 5-bromo-4-chloral-3-indol beta-D galactopyranoside. And so when the enzyme from the LAC-Z gene product was produced, it cleaved it, and the organic compound was cleaved, and it released indol, which is effectively the color of your genes, hence the term blue gene. I guess no one ever thought about it, but I thought it was important here because here the authors used Luciferase as their reporter, a favorite cassette often used by our guest and his wife to create portraits of various folk, Elio and Vincent, to name two. But the principle here is going to be the same. Drop the cassette into a transcriptional unit that can detect the presence of arsenic, hopefully at a concentration that would protect the drinking water of the general public. Now, there are numerous arsenic biosensors based on the regulatory system that came out of E. coli, or the ARSR regulatory system. And they're using reporter genes encoding either a light emission protein, the luciferase complex is a good example, or a fluorescent protein. The green fluorescent protein is often one of the most favorite reporter cassettes used today. Most of them have been hosted in E. coli cells, allowing arsenic detection in less than 1 hour with a high level of specificity and a detection limit as low as 13 nanometers or 0.1 grams per liter, which is 100 times below the threat level of 10 micrograms per liter. While the detectors in E. coli have come a long way, their commercialization and field use have not yet fully matured. So in this study, the authors introduce us to the use of the Magnetotactic Bacteria to be tailored as bioluminescence sensors to detect the presence of very low levels of arsenic. And the way they do this is they simply use magnets to effectively concentrate the bacteria. And if there's arsenic present, they will be able to detect the light because more of the bacteria will be present. They don't have to do a filtration step. They don't have to do a concentration step. All they need is a magnet. The bacteria will naturally migrate to the magnet.
Actually, they put the magnet to the side of the test tube, don't they?
And
pour out the rest of the liquid. And what's
yes,
in the test tube is magnetic bacteria.
That is correct. So what we're going to see is really a guide that these authors offer us and how they're able to exploit this unique group of microbes to extend the scope of applications to environmental monitoring and report that it's possible to modify two bacteria that they looked at. And the two that they looked at is Magnetospirillum magneticum and Magnetospirillum griffswaldensiae to facilitate the detection of arsenic-dependent expression of a transcriptional lux-based reporter gene. So at the bottom line, the study demonstrates a strong potential of magnetotactic bacteria to be able to address a very important public health question, which is simply, is there arsenic in our drinking water? And they, in the final aspect of their paper, they take us to really making this available for under-resourced countries. They assess whether or not they can freeze dry the sensor they create, resuspend it in water, and then simply ask the question, is the water got arsenic in it? So that's where we're going to go on our journey here today. So the first question the authors addressed is whether or not the magnetotactic strains could withstand arsenic. Again, because if arsenic kills them, the sensor wouldn't work. So they had already established that it's very easy to grow these two environmental strains. They were possible to genetically modify. And most importantly, they could grow and absorb metal ions such as cadmium, cobalt, manganese, and copper. Often, these other transition elements are often the general suspects when arsenic's around. Generally, these guys happen to be in the contaminated water as well. So they cleverly cultivated the growth of the magnetotactic bacteria in microtiter plates so they could monitor their growth in the presence of various concentrations of metal ions and, of course, arsenic. They learned that under these conditions, the resulting half maximal inhibitory concentration, or IC50, for arsenite was determined to be 259 micromolar for the first organism that they were looking at, the magnetotactic Aquaspirillum magneticum. And for the other one, it was 406. When the same experiment was performed on cultures of E. coli, it, however, had a much, much higher IC50 of 11.2 micromolar, millimolar, which was 2 orders of magnitude greater but appreciate that the concentrations that they were able to support the growth of their microbe was well over 2,000, and in the case of the second microbe, 3,000-fold higher than the toxic concentration present. So they were confident that they picked the right two bugs from which to build their sensor. So the next question that they focused on is they went on a hunt to determine if the two strains they were able to grow had an arsenic resistance operand. So here they resorted to good old molecular biology. They took the arsenic inducible promoter from E. coli, put it into the magnetotactic bacteria, and asked the question, we'll give it some arsenic, does it express? The answer was no. And that's principally because magnetotactic bacteria are so phylogenetically dissimilar from E. coli that it wasn't surprising that the promoter couldn't react to the arsenic because it was too different. So what's a good self-respecting molecular biologist to do? The next thing is they do an in silico analysis where they could dope their blast search parameters to ask, was there an operon in the genome of the magnetotactic bacteria that was similar to the Arsenite operons that we saw in E. coli that was using the ASR regulator system. They got lucky. They found 10 genes in microbe number 1, AMB1, and they found 8 genes in MSR, the other magnetotactic bacteria that belong to the ASR regulatory family of metal-binding transcriptional regulators. So they next needed to winnow down the 18 gene targets, which they did by analyzing neighboring genes, asking whether any of them were related to known arsenic resistance operons. And again, this is in silico. They found 3 putative arsenic operons, 1 in the AMB1 genome and 2 in the MSR. So from 18 down to 3. And their next task was to analyze which of the 3 targets would be most appropriate. And they did this by not the technique that I was taught, northern blots or dot blots. They did it with RT-PCR. And it was, again, a straightforward set of experiments that they were able to do. And at the same time, they wanted to know whether or not arsenic would inhibit the growth of these organisms. And so they tried 0, 50, and 200 micromolar concentrations. They asked the question, did it disrupt magnetism or growth? They were easily able to evaluate both phenotypes by looking at optical density and whether or not they were able to travel down magnetic force lines. And what they learned is that there was no significant impact by the arsenic, which they referred to as a metalloid ion, to either the fitness of the cell or its magnetic behavior. So again, great news if you're trying to build a sensor stain. In the figure where they present their RT-PCR data, they measured transcripts for the 3 operons between 0 and 30 hours subsequent to the addition of the signal, the arsenic. And again, think about your end goal. You're building a sensor. You want something that will remain upon exposure so that you can detect it for a long period of time. So condensing the data, with apologies to the folks who did all the hard work, they settled for the AMB1 reductase operon, where the induction of transcription is clearly observed after 60 minutes of exposure to either the 50 or 200 micromolar concentration of the signal. And since the second magnetotactic bacterium, MSR, grew faster under their culture conditions, their first sampling was made after 40 minutes of exposure. The take home for all of this beautiful work was that 2 reductase operons, arsenite induction, was maintained beyond 24 hours with an increase in the quantity of transcripts over time, and that the transcriptional experiments demonstrated that the promoters of the 3 operons identified by their in silico analysis were indeed regulated as a function of arsenic concentration, and they give a almost a staircase experiment where they increase the concentration of arsenic, and you can literally walk up the steps with them, again arguing that the regulatory elements that they have selected are indeed good candidates for the biosensor reporter gene system. As you might imagine, their next task was to construct the biosensors. Now here, they beautifully walk us through their construction project where they focus on the reductase operon and a methyltransferase operon to build 2 different sensors. They made a total of 4 plasmids. They describe how they do it, and they settled on Mark's favorite operon, the Lux CDABE operon, which requires expression of the whole operon, but it allows for the autonomous bioluminescence emission of the luciferase enzyme system without any need to add any substrate to the medium. So now this was many headlines news because autonomous luminescence reporting had never been used in magnetotactic bacteria, and it worked. So we're now 3/4 of a way through building their biosensor, but it doesn't mean we're out of the developmental woods. Next up is testing and specificity of their magnetic biosensors. And recall that sensitivity means true positive, that there is indeed arsenic present. So when arsenic is there, it's got to light up the operon. And specificity means true negative. So if there is no operon, if there is no arsenic present, the operon has to be truly off. But we know that all operons are a bit leaky, but theirs was sufficiently good that off was indeed off. So this is where they really begin to fine-tune things. So here they first characterize by monitoring the bioluminescence emission spectra over a range of arsenite concentrations in both strains of magnetotactic bacteria harboring the 2 different Lux-based reporter plasmids. And the response they saw for both strains was short, regardless of the promoter, resulting in significant signal only 30 minutes after the addition of the arsenite, which corresponds to one of the shortest detection times for an arsenic biosensor. Again, this is great news if you're building a sensor and screening lots of water samples. However, the sensitivities or true positives of their 4 biosensors differed significantly. And this is shown in their figure 3A and 3B. And for the biosensors built with the methyltransferase, they had significant luminescence signal was only measured for metalloid concentrations of 5 micromolar and 125 micromolar. And this is very problematic if your risk range is only 0.13 micromolar. So you built a great sensor, but it's not going to tell you anything unless the concentration is 1 order of magnitude above your threshold. The authors offered that this low sensitivity was in line with their low affinity of the arsenic transcription element for arsenic as hypothesized from the RT data that I didn't walk you all the way through. So if you have an opportunity to compare and contrast the RT PCR data to their functional assays here, you can actually appreciate the elegance of the work that these three authors really accomplished. But it's not all bad news. For as little as 0.5 micromolar arsenite can be detected in both strains, and the two biosensors designed using the AMB1 identified reductase operon. And they offer that the results demonstrate that the promoters selected from one strain can actually work in another strain. And that often happens. E. coli promoters can often function in other similar enteric bacteria. In addition, since the luminescence emission spectra was linear for both strains within a concentration of 0.5 to 5 micromolar arsenite, they can therefore be used in a semi-quantitative measurement of arsenite in environmental samples, either by fast detection in a lab or in the field using a portable luminometer. They also looked at the oxidation state of what turns the promoter on. So arsenic is its valence state is +3, and there's another form of arsenic in the water called arsenate, and it's got an oxidation state of +5. However, when they added arsenate to the medium, or the +5 valent molecule, it was not able to turn on the biosensor, and they attributed that to the fact that the magnetotactic bacteria don't take up the +5 component. It needs to be reduced to the +3 component before it can actually be taken up. So at the end of the day, they have a functional sensor in a microbe with a unique property. It will detect arsenic in the water as arsenite. Now, the beauty of the system is it's magnetotactic, so you can concentrate them into one spot using something as simple as Elio pointed out earlier as a magnetic stirring bar. And in fact, if you're looking for experiments to do with your students, they can actually go out to a stormwater retention pond behind their house, get a bucket of mud, lava mud to settle in. If they all have their magnetic stirring bar, they can actually see the little bugs swim out and attach themselves to the side of the jar. And you can actually look at them. You can literally watch them fly out of the pond scum, if you will, and able to do it. I know many people are looking for experiments people can do remotely, and a lot of us have stormwater retention ponds behind our homes, so you should be able to see whether or not, and magnetotactic bacteria are distributed all across the country, and they're associated with both freshwater and saltwater. So why did they care? For a while, they did indeed design a specific biosensor. Its sensitivity was not low enough to declare water safe to consume. So the lowest they could go was a 0.5 micromolar. And the WHO concentration is 0.13 micromolar for potable water. And for discharge from industrial plants, it's 0.32 micromolar. So you're not even getting to the level that industrial plants can discharge. So now you appreciate why magnets. To improve their detection limit, they exploited the magnetic character of the biosensor. They concentrate the bugs down, and this then allowed them to detect between 5 nanomolars to 1 micromolar arsenic in the presence of the magnet. And this easy-to-do magnetic concentration step increased the signal, and they were able to actually detect between 0.5 micromolar. They were able to detect at that 5 nanomolar to 1 micromolar concentration. And the final experiment that they offer us is they wanted to see whether or not they could indeed preserve the sensor so you could distribute it to get it to test water out in the field. And so here they offer us a freeze drying experiment. And freeze drying has always been an art. And they describe for us the different cryoprotections that they used. And cryoprotectants that are typically used are things like sucrose, dehydrated or powdered milk, DMSO, mannose, or trehalose. And so they use 2 concentrations of magnetotactic bacteria, either at 10 to the 11th or 2 times 10 to the 11th. They mixed them with the various cryoprotectants, and they simply revived them by adding water to their aliquots. And they learned that for the conditions where mannose and trehalose were used as the protectants, the biosensors were capable of giving a luminescent signal after incubation with arsenite at concentrations above 1 micromolar. And again, you can do a concentration step where you can actually effectively get more signal into the area to improve your sensitivity. They ultimately settled on trehalose for the freeze drying at a final concentration of 50 grams per liter and incubated the magnetotactic bacteria at room temperature, which even gives them greater utility for field use rather than at the defined optimal temperature for growing these magnetotactic bacteria at 28 degrees. So this was a complete story from beginning to end using magnetotactic bacteria to make a biosensor, really effectively teaching us how if we can concentrate the signal, we may be able to detect a condition above the noise. So that's it.
So, Michael, let me ask you the obvious question. Do you think that the magnetotactic bacteria can be used to detect other things, like the coronavirus? It takes some engineering, but it seems to me that it sounds feasible to make magnetotactic bacteria sensitive to all kinds of things.
Absolutely, Elio, and I think that's why I pulled this paper. I was biased when I was doing my hunt last week. I had just listened to Minna's incredible discussion of cycle threshold values on TWiM. What was it, 640, Vincent?
TWiV
640. I'm trying to appropriate TWiV to TWiM.
Yeah. Okay, it's all one.
It's all one. But he did such a fantastic job. And he talked about pooling samples. The Red Cross routinely pools blood samples to screen many more blood samples with less resources. And this is a way we could effectively do it. And I think if we build the spike protein into a cassette that has luciferase and puts it into magnetotactic bacteria, and we know the right signal to set it off, we could effectively detect it, or the receptor that the spike protein uses.
So you're going to put the receptor in the bacteria. Is that what you're saying?
Yes.
And it's going to be produced, then it has to be put on the cell surface, right?
But magnetotactic bacteria are good God-fearing gram-negatives, so they do export.
So you get the ACE2 receptor on the surface, and then if there's some spike floating around, you pick it up.
Yes.
Right? It's just going to bind. It's going to be a little bit different of an assay here than here, right?
Well, we could make a fusion so that when it does bind, we could do it with a coupled G protein type thing where when it binds, it does a kinase reaction and then turns on the luciferase.
So you could certainly do a 2-component system version of that.
Yes.
Exactly. No, I think you could do. The thing is that a strip impregnated with antibodies is far simpler, right?
Oh, yes. Minna had it nailed. And I want those strips to be distributed to every parent in South Carolina so we can send our kids back to school.
I mean, everyone has to listen to TWiV 640 if you haven't, because this is a revolutionary. I mean, it's so simple and it just changes the whole game.
And the FDA approved this test strip in May of 2020.
Mm-hmm.
I think that's in the show notes. It was approved in May of 2020. And the question is, as Dr. Minna so eloquently say, why hasn't the government started printing these in mass?
Yep, that's right.
I think that's a really good idea, and to try it out in some places, like perhaps a small undergraduate institution.
Yes.
Oh, totally. You get every student loaded up with them, and before they come to class, they lick it, and if they're positive, they stay in their dorm room for 10 days. CDC says 14, but listening to Minna, you're not transmissible for more than 7 days.
Yeah. Well, the Nature paper showed that, and that was published in mid-April. So we have a tremendous amount of data out there. And so that was my madness in picking this particular paper to walk us through of how to build a biosensor.
Is this going to be used, you think, Michael?
I do, because arsenic poisoning is huge. It's a substantial issue. These under-resourced countries have a rough time, and arsenic poisoning, there's no easy therapy short of chelation, which is very medically intensive.
Can I make a couple of comments, please?
Yes,
please.
Yeah, the first thing I wanted to say is I hope you've all seen Manu Prakash's kind of super one-step SARS-CoV-2 detection thing that involves little hand spinners. If you haven't, I'm happy to add a link to it. I mean, that lab up at Stanford is just superb at low-cost things that are going to be used all over the world.
This is the guy who made the paper microscope.
Indeed. It's something called a LAMP procedure that I'm learning about. And I think it's an interesting thing that he's already published on. But getting back to your paper, Michael, this is a big deal to me because just about 2 or 3 miles from here, we used to have a smelting plant here in Tacoma called the ASARCO plant.
Oh, my.
So there are lots of heavy metals and lots of toxicity in what would you call when you get landfill? That's the word I'm looking for.
Landfill runoff. Runoff.
I have been interested in finding a heavy metal responsive promoter to use for exactly this purpose. So I'm wondering if I can like wheedle these people into sharing the strain to use it, because what a great thing.
It's in AEM. That's one of the requirements of ASM journals.
So just 2 more comments, because this is kind of a little bit in my wheelhouse. I couldn't find what the source of their Lux cassette was. My guess is it's from Photorhabdus. And the reason I say that's the only terrestrial bioluminescent bacterium. So that's an interesting bit. I can probably track it down, but it wasn't easy to find. Certainly it's the intact cassette rather than just Lux AB, where you have to add an endogenous aldehyde to make it work. And then the other thing that I wanted to say about this, and this is what really excited me. Yes, finding out about dealing about arsenic poisoning, very important. In no way am I negative about that. But I have wondered for years about using Luciferase as a reporter gene under low oxygen environments. And you've just demonstrated that you can do that.
Mm-hmm.
Because the magnetotactic bacteria are microaerophilic, and they will not make magnetite if there's a lot of oxygen around.
That is correct.
So that means that you can actually use this under low oxygen conditions, because if you look at the bacterial Lux reaction, just like every Lux reaction on the planet, it requires oxygen. And so I would have thought this is not a good choice, and I would have been wrong.
You would have bet on the fluorescence molecule.
Yes. And even then you have problems, because under certain conditions with low oxygen, things don't fold properly, so on and so forth. So this is really quite remarkable stuff.
Yeah, what they say in their methods is that the Lux CD ABE operon was inserted using speed 1 digestion, and that's all that they offer.
Yeah. So my guess is they're using the Photorhabdus version, because a lot of people have been using it. I am not sure how the marine bacterial structures work. That would be Vibrio fischeri, also called Ali Vibrio fischeri, apparently. They keep changing the names on me, Elio. I know.
Don't tell me.
Oh. So anyway, that appears in one relatively conserved operon that could be easily put in there, but it also does it in photorhabdus, and a lot of reporters are using it. So that's my guess. I've actually used some of their base plasmids in the past, and they're excellent.
Mark, is this something you could do with your students to go check the runoff?
Yes, 1,000 times yes. By the way, you can make a super cheap luminometer. I'm trying to help a person over in Europe for like a high school project to measure light. And there's apparently some things you find on the do-it-yourself websites about making a very, very simple photometer, which is all you would really need for this. You wouldn't want to use an expensive one. My luminometer is my favorite piece of equipment because you put the tube in it, you close the top, and you hit go.
And you get a value.
And you get a value.
Great. Well, I have one other comment. This would be a good case for a graphical abstract. So I want to say, ASM, get some graphical abstracts in your journal articles. Don't you think one showing the magnet and the bacteria and all of that would be great here? I know they don't want to do it because it costs money.
But it doesn't. It's just electrons.
Well, if you have the authors draw it, that's fine. Right. But if you have some professional draw, I don't know what the, cell journals do, whether they have authors submitted or what. But I can tell you some ways that ASM could save money, right, Michael and Mark? Elio? Yeah, we do know that. All right. Thank you, Michael.
Thank you.
Our last bit for today, I want to read one letter from Jane's, all about fluoride.
Oh, my.
Regarding last episode 217, you were talking about the benefits of water fluoridation. I do not know to what concentration levels the authorities add fluoride to the drinking water in America. The so-called DMFT, decayed, missing or filled teeth index, if I recall correctly, is not much better in the U.S. than over here in Central Europe, where we do not add fluoride to the tap water, which is a good thing, I strongly believe. I surely do understand the chemistry behind making the enamel more resistant to lower pH by swapping the hydroxyl with a fluoride. However, forbidding the use of high fructose corn syrup and all industrial produced sucrose would have a much better effect on dental health than fluoridation of tap water. So what is more acceptable? Would a tax on sugar be a good compromise to keep the Stevens curve at dental-friendly levels? It would be considered unethical and against the law here to add anything to the tap water, however beneficial it might be to some people who do not take care of their alimentation, which is the ultimate cause of making oral bacteria sweat acids. Strep mutans is not always found in carious lesions. Processed food and sucrose, dextrose everywhere in amounts not possible before the advent of industrialized food production cause an ecological environmental shift in the bacterial composition in the oral cavity microbiome, as explained in the last episode. When these bacteria attach to the teeth and form a biofilm, they will be starting to produce acids if one continues to eat a lot of industrial foods, concentrated sucrose. Strep mutans is not required for this process, and he gives a link. Marsh Oral Microbiology, caries management, and caries excavation. People from areas where you do not find a lot of processed food sugar tend to have excellent teeth until they arrive here. The dentist Weston Price documented in his book Nutrition and Physical Degeneration that we just need to brush our teeth because we eat what we eat in the Western world. Maybe there is a chance to get some experts on the show regarding this topic. Tooth decay is one of the most common health problems, and if the circa 1,000 different bacterial species in our mouth enter the pulp and thus our blood system, you will see patients coming up with heart problems. There certainly has been a paradigm shift in the understanding of dental caries, or ultimately which bacteria sweat acids and why, but I do not follow the Journal of Dental Research so closely anymore. One thing which is puzzling is that secondary caries below fillings is possible, as it must be because of enough substrate, enough sucrose finding its way below the filling microleakage. As described in Schwendick's book, not all of the bacteria are removed anymore when excavating close to the pulp in order to preserve it. Seal is the new deal, and studies how this plays out are ongoing. Another interesting topic for a podcast would be the known relations between tooth decay and heart disease, and he sends a link for that. Wish you all the best, and thank you for your great work you do. I always love learning this way. Any thoughts, Michael?
Well, Mayor Bloomberg can comment as to a sugar tax, and you live in New York City, Vincent. So How has the sugar tax been received?
That wasn't done, was it? I mean, he wanted to tax the big, the supersized soda, right?
Which is effectively a sugar tax.
Yeah, and they said no, you can't do it.
Yep. So I don't think that's going to happen. We fortunately were able to get fluoride into our drinking water because during World War II, so many of the draftees were 4F because of their bad teeth.
Yeah.
And consequently, that was what was the impetus for really fluoridating water, because it was fairly inexpensive, it was easy to do, and it actually worked.
There's no side effects of putting fluorine in the water, right?
No.
Mm-hmm.
No.
All right. One last one from Justin. Dentists extract new fee from patients to keep up with rising COVID-19 costs, and provides a link. And he writes, pretty sure TWiM said this a month ago. Oh, yeah, the costs are going to be passed on to you. I got a letter from TWiV. I think Wesleyan University up in Connecticut is charging $16,000 extra per student per semester.
Holy moly.
All of the precautions they need to take, disinfection,
plexiglass,
whatever. And I just think this is terrible. I think the colleges need to pay up, as I've criticized on TWiV. Cornell, they don't want to test. They don't have the resources. Baloney. Don't charge the students.
Well, they have to come up with clever ways of testing. And, the whole issue, the Red Cross figured out how to economize so they could still preserve the safety of the blood supply. So they pool blood samples. Yeah. And they can assess whether or not they have any communicable diseases in the blood. And if it comes up positive, then they go back and individually test each bag before it's released. And similarly, we could do that. We have the technology. It's fast enough if it's done often enough. And I think that's the key. It's got to be done often enough, because Tetris does work, but it only works if you test often enough.
Yep. All right. That does it for TWiM 223. You can find the show notes at asm.org/TWiM. If you have questions or comments, you can send them along to twim@microbe.tv. And as I said before, if you like what we do, you can support us. Go over to microbe.tv/contribute. A couple of different ways you can do that. Today on TWiM, Elio Schaechter. You can find him at Small Things Considered. Thank you, Elio.
My pleasure. Thank you.
Michael Schmidt is at the Medical University of what?
South Carolina, where we have the world's record of COVID going on right now, today.
We have more cases than anybody else?
Per capita. It's unfortunate that we have continued our misbehavior of not wearing masks when out in public.
Oh, it's a dubious honor, Michael. Well, anyway, thank you very much for joining us today. Mark Martin's a professor at the University of Puget Sound, which is in Tacoma, Washington. Mark, thank you for joining us today.
It's been a real pleasure. Thank you all.
Come back again.
I'd love to.
We'll make you more famous than you already are.
I'd rather be infamous than famous, that kind of thing.
That's right.
Yeah.
You're doing fine, Mark, and we really appreciate it.
Yeah, thank you.
Thank you, Elio.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank ASM for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.